great to virtually meet everyone. Pleasure to be part of this NYSF seminar series. Um, let's hope that my recording technology will work okay. So hello again. My name is Audrey. I'm a new PhD candidate at the ANU. I've actually just started my degree February 2020, so about to be 3 months into my degree, which is coincidentally about the time for my project milestones to be due. I did a Bachelor of Science honors before I did my PhD. I had a 1-year gap, and I work at the Moritz lab. So it's led by Craig Moritz here, which is my primary supervisor. Most of the work in our lab focuses on phylogenetics and biogeography, and really the best way to describe everyone in our lab is that we love evolutionary trees. We love maps, graph, and results in general. Craig and the lab does a lot of work with geckos. He actually has one named after him. And I actually first started working with him on a 3rd-year project where we named a bunch of new species of Gahaira geckos. I have a co-supervisor for my project from the CSIRO, Leo Joseph. He does a lot of work with birds in Papua New Guinea, which will make sense later when I explain what my project is. A lot of my CSIRO involvement will be access to the Australian National Wildlife Collection, where they have one of the largest and very nicely organized collection of bird samples and tissue samples in Australia. As I'm in the very beginning of my project, a lot of my current work now revolves around reading around the current literature, finding the gaps in the knowledge, and really the most important part is synthesizing the questions that I'll be asking for my project. So in this presentation, I'll be mostly talking through my thought process on what I currently have in my project, which is tentatively titled Historical Biogeography and Avian Speciation in the Indo-Australian region, or what is also known as the Indo-Australian Archipelago, or the IAA for short. Simply put, to sort of basically summarize what my project will be, I want to look at how the dynamics of geological history, such as the movements of continental plates and the emergence of submergence of land masses, creates a unique environment for the dispersal and speciation of bird species in the Indo-Australian region. But first of all, why the Indo-Australian region? Well, aside from my personal biases, for myself being Indonesian and living in Australia for the last 5+ years, the Indo-Australian region is considered one of the largest hotspots of biodiversity. The whole region itself occupies only 4% of terrestrial habitat globally, so it's quite small in terms of percentage. However, it is home to nearly 25% of the current living terrestrial species. Indonesia themselves, about 15% of all existing birds in the world are found in Indonesia, so that's around 1,700 species out of the 11,000 that we know of currently. Not to mention, both Indonesia and Australia, and also and the area in the IAA, have a large proportion of species that are highly endemic to the area, which means they are not found anywhere else in the world. Other than that, the Indo-Australian region also has a very interesting biogeographical history. Now, biogeography is the study of the distribution of species across a spatial range. Here we can see a map of the IAA, so Australia here, Indonesia, and then we have Papua New Guinea. A lot of people divide this map into what we 3 major parts of the IIA. The west makes up the Sunda Shelf, so this is mainland Asia and the western part of Indonesia. The east, the eastern Sahul Shelf, which is Australia and New Guinea, and the islands in between. Now, this area in between we call Wallacea, for reasons that I will explain later. So Sunda and Sahul, they're what we call continental shelves. Some interesting to note, they used to be connected inland, which means mainland Asia used to be connected to this whole grayed out area of Sunda, and New Guinea and Australia was connected as well. So we can see it used to be a pretty big land masses. However, interesting to note, they have never been connected to each other. So there's always been this islands in between have never been connected. And that's sort of why we'll see later, Wallacea is one of the most interesting part of the IA. Now, we can see there's a couple of lines here as well, though these are what we call biogeographical lines. And one of the most important and famous one is Wallace. Some of you may have heard of the Wallace line. It's quite significantly famous. And it's what we know as a faunal boundary line, which is drawn by the famous naturalist Alfred Russel Wallace. He observed that organisms to the west of the line, so to the west of the line here, in mostly Sunda, are more related to Asiatic species or Asian species. And to the east of the line, they're more related to Australian species. So here we can find the eucalypts and the monotremes, kangaroos, and to the west we can find tigers, rhinoceros, and interestingly, his observations hold up with current research. So a lot of results are consistent with it on a large number of species, including birds. So birds, some bird species can't actually cross the line. They're only found on the western side or the eastern side, such as woodpeckers. And it's very interesting because if we think about birds, they can easily fly. And the distance between Bali and Lombok, which is here in this little area over here, is only 32 kilometers. So why can't birds fly across it? And the simple answer to that is yes, they can actually cross it. And some species do this very successfully, such as parrots and songbirds. Parrots and songbirds are found throughout the world, even though they originated from Australia. So what is different about these birds that can or can't cross the line? What is so significant about this boundary, which is the Wallace line? What makes it, aside from the fact that it's a large, deep ocean trench, that it shouldn't hinder birds from being able to fly over it? These gaps in the knowledge would be the basis of the question I will ask on my PhD. So the first main question that I will be looking at, which will be the foundation for the rest of my work, is where did the birds in the IA region originate from? So there's many different possibilities. In the past, continents have moved around. Some islands act as stepping stones, but really we want to describe the bird diversity in the region. What groups and families are currently present? Are there specific features that stand out for groups that are found only on one side of the Wallace line? Or are there features that are shared if they can cross the line? So these are the sort of starting point questions that I'm trying to look at. Secondly, would be the habitats they're from. In the past, what kind of habitats did they come from? Because a different range of habitats are found in the region. Whilst Indonesia and Papua New Guinea are mostly moist evergreen rainforests and montane rainforests. We have mangrove ecosystems. It's a tropical equatorial area, and Australia is largely arid and temperate. Although these biomes and habitats have changed from the past, but the question that we can ask is, did current savanna species in the region, did they evolve from savanna species of the past, or did they originate from rainforest species that adapt to new environments because of a new opening? And to address this question, I will be looking for evidence of what we call biome shifts, which are changes to a species' preference in their environment. So how do I look for these biome shifts? I'll be using what we call a technique called ancestral range reconstruction, where I'll try to reconstruct the geographic range of the species' ancestor in the past. And we'll see how that compares using genetic data and using biome data, the current biome data, and we'll see how that compares to what we know of the current distribution now. Lastly, what pathway did these birds take to colonize Australia? Because first, how did the birds cross from the Sunda Shelf to the Sahul Shelf? Did they go through Papua New Guinea? They jump islands. Do they use islands as stepping stones, or did they fly from Malaysia directly to Australia? Why can some birds do this but not others? And what features do they share amongst the group? Secondly, between Papua New Guinea and Australia, where they used to be connected, there are different habitat types in this small crossing. Which path did the birds take, and how does this differ between groups? So these are just some of the starting questions I'm trying to understand in my PhD. There's other aspects to it. Oops. There are other aspects of it, mainly on bird coevolution with viruses that we may touch on as well as a final section of my PhD, but that's a story for another time. And for now, by combining ancestral range reconstruction, current distribution, phylogenetics, and our knowledge of the geological history of the region, I hope to be able to shed some light in answering exactly how did the historical environment of the IIA region create the unique biodiversity that exists there today. Thank you very much, and hope this has been as interesting to you as it is to me. Cheers. Bye.
Hi everyone, I'm Jinjia. I'm from Research School of Biology in ANU. Today I'm going to introduce my PhD research, which is about sexual differences in morphology and behaviors in fiddler crabs. Before we get started, let's have a look at some photos, and it's a small activities just to try to guess which is male and which one is female. The first species, I'm pretty sure it's very easy for all of us. Second species, this is fiddler crabs. And the turkey, elephant seal. Okay, this is the answer. So as we can see, males and females sometimes can be very different in their morphologies, and this is actually really common to observe in nature. We call these phenomena sexual dimorphisms. This kind of sexual differences in the same species between two sexes actually triggers a lot of questions and a lot of curiosities in evolutionary biologies. And that's because these two different sexes, they actually share the same genome. But why and how can they express their trait and sometimes even their behaviors in so many different ways? So we really want to know that where are the differences from? And there have been some classic studies trying to explain this phenomena. They hypothesize that these kind of sexual differences is actually from the respective reproductive roles played by the two sexes. For example, one of the most fundamental differences between two sexes is actually the reproductive materials produced by them. So we all know that females produce eggs while males produce sperm in reproduction. And because eggs are larger, are more nutritious than sperm, so this actually indicates that females, they invest more in parental cares in reproduction. Females not only invest more in the eggs, but also engage in a lot of parental cares after mating. For example, they might have to produce the offspring directly, or they sometimes have to carry the fertilized eggs and even incubate them. After hatching, they might have to search for the food and feed the larva. So actually, this kind of inequality in parental investment can be quite obvious in a lot of species, and usually females are the size that invest more in parental care. So since females are usually the side that put in more effort and energy in taking care of the offspring, the limitation of the female reproductive success now will be the nutrition that she can acquire to produce the eggs or to feed the offspring during reproduction. And also, since she is a size that often invests more in parental care, so she must be really careful about who she wants to mate with, who she wants to partner with, so that she can make sure her investment is meaningful, she can raise the maximum number of the offspring within her capacity, and also the partner can benefit her from increasing the offspring fitness. So usually, females are more choosy in mate choice. On the other hand, male invest relatively less in parental investment, so nutrition will not be the major limitation of his reproductive success. Instead, the mating partner will be the limitation of male reproductive success. So the way to increase their reproductive success is to mate as many times as he can so that he can be the father of as many offspring as possible. So these might be the reasons that we can see a lot of exaggerated traits and fancy courtships that have been evolved in males. In some of the species, male monopolizes female partners through winning a fight and claiming his territories, while in other species, the males try to convince the female males that they can be a good father by displaying a really fancy courtship. So now we've already have a more clear idea about where the sexual differences in morphology and behaviors are from. And one of my PhD research aim is that we want to further understand that what are the mechanisms that help to maintain this kind of sexual differences. For example, the story we just mentioned is constructed based on the fundamental assumption that females have greater parental investment and thus she had higher nutritional demands. So to support the egg production, does she really eat more or she just allocates more nutrition in the reproductive organs? I address these research questions using fiddler crabs Euchar borealis. Fiddler crabs is actually an ideal study system for these research questions. As you can see that males only have one feeding clothes, and the other clothes is specialized to display courtship or engaging in territorial competitions. On the other hand, females has two feeding clothes. So these kind of sexual differences in morphology can further result in a different feeding behaviors, which makes them an ideal study systems for addressing sexual differences in nutritional processes. We first want to know that if females eat more, and we look at the feeding rates. What we found is that females actually feed faster, have higher feeding rates than males. This is actually making sense because females have two feeding clothes and they are using both of the feeding clothes in feeding. Second, we want to know that if male and females have different size of the feeding clothes, and this time we found that males actually have larger size of their feeding clothes. We also want to know that who spent more time on feeding, and as you can see here that the number of males are pretty stable throughout the time, so it suggests that males spent longer throughout the day on feeding. So do females eat more? We think the answer is no, and we think that in Euchar borealis, males and females are likely to have similar amount of food intake. Although we observe that females feed faster, females have higher feeding rate, the male can be compensated the loss by having a larger feeding clothes or spending more time on feeding. If females don't eat more, do they allocate more nutrition in gonads? To answer these questions, I carry out a stable isotope experiment. The stable isotopes were sprayed to these sample areas. I wait for the assimilation of these enhanced isotope signals to the crab bodies, and then I capture the crabs, dissect the tissues, the gonad tissue here, and see how they allocate these nutrients by chemical analysis. We first found that females actually allocate more delta C13s into their gonads compared with males, and this kind of differences can be quite significant. We also found that females actually allocate more delta 15N into their gonads compared with males, and this kind of differences is also significant. So, do females allocate more nutrition in gonads? We think the answer is yes. Eukarborrelate Alice females actually allocate more organic carbons and nitrogens into their gonads. This study presented direct evidence supporting the hypothesis that females actually have a greater pre-mating investment. We further explained that nutritional allocation is the major process supporting sexual differences in parental investment in fiddler crabs. At the end of this presentation, I would like to thank ANU for providing the travel funding and research funding. I would also like to thank all my volunteers, all my friends, collaborators in Hong Kong for helping me through data collections and analysis in Hong Kong. Thank you.
Hi, I'm Brittany, and I'm in the third year of my PhD in the Fenner School on Ngunnawal land at the ANU. As you can see, I am pretty much never happier than when I have a cute animal in my hands, which is just the stereotype of the ecologist I am. But I break that stereotype a little bit in that I also studied genetics to help me conserve our wildlife. My thesis is part of the Mulligan's Flat Garuyuru woodland experiment. I'm using 3 case studies, the eastern bettong, eastern quoll, and New Holland mouse, to help me look at what constitutes the effective use of genetics in reintroduction biology. As you've probably already guessed, conservation genetics is where you apply genetic analyses to conservation projects. This used to be a bit of a fringe science in ecology, but it's becoming more and more accepted and more popular. And we're actually starting to have specific disciplines forming. And reintroduction biology is one where conservation genetics is starting to take off, and that is where my thesis is focused. Reintroduction biology very specifically looks to offset species decline using translocations to supplement and even reintroduce populations that have undergone reductions and local extinctions. There are some inherent issues when you're moving animals around in this way, and there are logistics, some that arise from complex species interactions, and some that are due to drivers of decline being hard to mitigate. The difficulties that are involved specifically with establishing these populations are where conservation genetics can help out. The Mulligan's Flat Garuyuru woodland experiment has been testing what does and doesn't work for ecosystem restoration for quite a while now. They're looking to restore a critically endangered boxgum grassy woodland, and they've been undertaking robust ecological and genetic monitoring ever since they started. It's surprisingly uncommon for reintroductions like that of the bettong, the quoll, and the New Holland mice to have such good genetic monitoring, and that gives me a really rare and lucky opportunity to look at the genetics and trends across time rather than just a snapshot. I also just want to acknowledge that it's the collaboration between all of the researchers involved in the MFGO, of which these are some of the current ones, that really helps this project be effective. And I'm really lucky that I can compare my genetic data to their ecological studies to help us really understand exactly what's happening and what is driving the success of our populations. When I'm thinking about genetic considerations for reintroductions, there are 3 broad areas I'm keeping in mind, and these are inbreeding, where related individuals are crossed together, and this can result in inbreeding depression and negative population level consequences. There's outbreeding, where if we've brought together individuals that are too dissimilar, the combination of their genetics can lead to bad things for the population. Then there's a general genetic diversity, which is a really important metric for adaptive potential and helping the population be successful. First off, I just want to say when I'm thinking about all those things, I'm thinking about data that I've gained from single nucleotide polymorphisms. A SNP is a site where one base pair has been replaced by another, which means we can only ever have two versions of an allele. But we do get thousands of these and they span the entire genome, and this gives us a really good data set. In diploid species like mammals, we each have two chromosomes, and so we have two copies of a locus. So the locus can have the same allele on each chromosome and be homozygous, or they can be different on each chromosome and be heterozygous. And heterozygosity is a common metric to talk about genetic diversity. And increased homozygosity is actually something that's often linked to increased levels of inbreeding. You can't entirely separate diversity stats from inbreeding ones because increased inbreeding leads to increased homozygosity because related parents with more similar genomes are crossing together. And this really famous example below shows that when the Florida panther became more and more inbred, more and more homozygosity occurred, and this included the expression of a deleterious allele for a kinked tail. And actually, they knew that these kinked tails existed for a long time, but until they did genetic analysis, they didn't realize that it was an expression of something that actually had really negative consequences for the population in the long term. And that's why we have to analyze genetics. And we also have to think about diversity in terms of adaptive and evolutionary potential, because when we're moving individuals from one location to another, they could encounter new things. They really need to be able to adapt, and so they need diversity for evolution to act upon. And this is something we have to keep in mind when we're selecting founders, because we know when we take a small group of individuals from one population and create a new population with it, we are putting them through what we call a genetic bottleneck. To help offset this, we try and take individuals from lots of populations, and this means that when we put all of their genetics together, even though we've individually bottlenecked each parent population, the reintroduction occurs with a high level of genetic diversity. And this was something we did for the eastern bettong. I have been thinking about inbreeding, outbreeding, and genetic diversity for this population. And I want to be able to answer some really important questions about what's happening in these reserves so we can adapt our management. When I wanted to look at whether or not we created a population similar to those that occur in Tasmania, I found that the populations we'd created in Mulligans and Tidbinbilla actually couldn't be differentiated based on where they came from. And that is really exciting because it means we've created a pan-mictic population. So all of the individuals are breeding together successfully. To look at this at an individual level, I used a program called Structure. So each source population is represented here as numbers 1 and then 4 to 12, whereas the reintroduced populations are groups 2 and 3. Each of these colourful lines represents an individual, and as you can see, the sources are groups of one colour, whereas our reintroduced individuals have lots of different colours in them, and this shows us that each individual is a result of breeding between different source populations, which means each individual is likely to be highly diverse. For logistical reasons, we want to know if we can use these new pan-mictic reintroduced mainland individuals as a source for future translocations. To do that well, you need to do quite a lot of modelling and advanced statistics, but we can think about it in terms of diversity we gain from the actual founders to what we could gain from the current mainland populations. You can see here that if we take the same number of individuals from the mainland as we did back in the day from Tasmania, we get diversity stats that are really, really similar. So for heterozygosity and inbreeding levels, everything is looking great. It's only when we look at some of these stats that reflect differentiation that we get a big difference. And we do expect that difference actually, because as we saw back in the structure plot, each of those source populations was really different to each other and similar to themselves. whereas our reintroduced mainland populations are a mix of all of those sources, and so are obviously less different. And so keeping all of that in mind, I can look at the reintroduction we've done for the eastern bettong and answer some pretty important questions just using genetic data. I can say that we have created a population similar to what exists in Tasmania, but each individual is actually more diverse than any individual from a single source. And I can say that these populations look like they are set up for long-term success with high levels of genetic diversity and low levels of inbreeding. And they can be used as a source for future translocations, which saves us quite a lot of time and money for any future translocations. And what is extra exciting is that I can take the results I found for the bettongs and apply it to other projects. I can give advice on how to obtain genetic diversity from source populations and how we should be thinking about genetics for reintroductions, and also how we should be adapting management based on genetic analysis to help improve a population's genetic health. And that's really exciting. Finally, I just want to say that no matter what you end up studying, just make sure you're passionate about it. You'll be able to produce research that's really meaningful to you, which I think is one of the best things a scientist can do.
Hi everybody, I'm Ollie. I'm a PhD student at the ANU, and this is the title of my talk, very broadly titled Evolutionary Genetics, Population Biology and Conservation. As an initial disclaimer, I'm going to say that today I won't be able to talk about the specifics of my results, and that's because at the moment they're pretty preliminary and they need validation. They don't really form a clear picture. You can ask me at the Q&A and I'll answer in as much detail as I'm able based on what I have, but it's not really presentable. So what I will be talking about is I hope to give you as broad an overview as possible of the project that I'm currently working on by way of my study system, what makes it interesting, how we collect our data, what our data looks like, and then what kinds of questions we actually ask with those data and why they're useful in this particular context. So I guess let's get started. So let's start with some conceptual background, just to give you an idea of the kinds of biological processes that we're talking about. This is a 3-species tree, and just by looking at it, it's pretty easy to understand the evolutionary relationships between these 3 species. Obviously, if we added more into the mix, it would get more complicated, but as it is, it's pretty simple to describe in the broad sense how these species relate to each other. What's not simple is if we want to understand in more detail. We want to, for example, see what happens within and not just between species. So here, each species will probably have multiple populations that themselves can also be described by some kind of tree-like process with one branch splitting into 2, those branches split, and then so on. Basically, what I want to impress on you with this slide is that a simple species history can belie a very complex population history. And it's at this level of biological organization that I'm mostly interested in populations. So very broadly, how can we study evolutionary processes in historical populations to understand what's happening today? So I study historical populations. This is my study species, the Lord Howe Island stick insect. It's a reasonably large, about 10 centimeter nocturnal flightless stick insect that was once abundant on Lord Howe Island in the South Pacific, but about 100 years ago it was driven extinct by the accidental introduction of rats to the island. There's only one remaining population known today, and it's on Ball's Pyramid, about 20 k's off the coast of Lord Howe. This is a very small, very restricted population, which by definition makes it critically endangered. And there's a conservation program centered around this species run by Zoos Victoria that aims to take individuals from the pyramid, breed them in captivity, build up some numbers, and then reintroduce those individuals to Lord Howe Island after the eradication of rats. This is obviously a really exciting idea, a really exciting prospect, but it does raise some tricky evolutionary questions. Now, here's another tree diagram. They pop up a lot in biology. Just a reminder that as we move from the tips, so down near the islands, the ends of these lines up towards the top of the screen, we're moving backwards in time. So at some point in the past, there was a large ancestral population of these stick insects that probably lived in the same geographical area, just when sea levels were lower. Then, a little later, at some later time in the past, the population split and came to occupy just the two islands that it occupies in the modern era. Now, obviously, the Lord Howe Island population is extinct, like right now, today, but we would still consider it a modern population in the evolutionary sense. On an evolutionary timescale, 100 years ago isn't really that long ago. So after they split, it's probably fair to assume that the two populations didn't exchange any migrants. There were no individuals moving between the islands because the insects don't fly, they're not very mobile. And so because of that, the two populations probably evolved along independent evolutionary trajectories. So they'll have accumulated different sets of random mutations, and they might also have accumulated different sets of adaptations to their respective islands. And this has really obvious conservation implications, I think. If an individual from Ball's Pyramid is adapted to Ball's Pyramid, we take that individual and try to reintroduce it to Lord Howe, the probability of that population actually establishing on Lord Howe is going to be lower than we expect. And with that in mind, the primary aim of this research is to study genetic diversity and adaptation in the extinct and extant populations to give context to the reintroduction. Here we hit a pretty obvious difficulty. The extinct population is extinct. They're no longer there. Fortunately, it's a very large insect that was abundant on Lord Howe before its extinction, so there are quite a few specimens preserved across Australia's museum collections from that original population. And it's not only possible, but fairly common nowadays to extract DNA sequence data from museum specimens. So these days, when we want to get DNA sequence data, we'll use what we call short-read sequencing. That's the most popular technology. So instead of trying to sequence a whole long DNA molecule at once, like a chromosome, we'll cut that molecule up into lots of short fragments and then sequence those fragments. Then we use computer algorithms to compare those fragments and line them up against each other, and in that way reconstruct what the original molecule looks like. So on the left is a screenshot from a piece of software that I use just to give you some visual intuition for how this process works. Now, each one of those colored greenish or yellow horizontal lines is a DNA fragment that's been sequenced from a sample. And what you're seeing is their alignment. So they've all been lined up against each other based on similarity. And those highlighted regions are differences between some of them. And by taking the consensus at each position of all of these lined up fragments, we can figure out what the DNA in the sample was like. So why this works for museum samples is because the DNA in museum samples is already fragmented, it's been degraded. And this meant that it used to not be usable because older technologies relied on having longer fragments. But now that short-read sequencing is the most popular form of DNA sequencing technology, museum samples are being used a lot more often, just because now we actually can. Now, this is an example of what the outcome of that process looks like. What we ideally want to end up with is a collection of sequences from several individuals per population. And then we line those up against the reference and against each other, as I've done here. We can start looking for differences between individuals and between populations. Once we have those, we can start asking questions about what produces those differences. So what evolutionary processes have led the DNA sequence to evolve in this way. We can start pretty far back in time and ask questions about the ancestral population. It's often useful to try and estimate how large this population was and how much genetic diversity there was, because there are some evolutionary processes that depend quite heavily on what the ancestral population was like, not just what it's like today. Moving forward in time now, we can start to ask questions about the split event. So things we might want to know are exactly when it happened and how many individuals there were on either side of that split, because that can inform us about how we expect evolution to proceed in the populations after the split. And then after that, we can ask questions about how evolution proceeded in the two populations after they split. One particularly interesting question is how fast DNA sequence evolution appears to be happening in these two populations. If we find, for instance, that the sequence is evolving faster in one population than another, we might then ask, well, why is that happening? What is the population experiencing to make evolution proceed at that rate? And finally, we might just want to ask questions about the present day. So the genetic diversity in the present day is one of the things that informs our answers about all of the past processes that I've just discussed. The present day is also where we would look for signals of adaptation in the DNA sequence data. Now, obviously, there are a whole lot more questions we could ask, and we could ask each of these questions in greater detail and about specific hypotheses that we have. But this has just been a whirlwind tour of the sorts of questions we can ask when we have DNA sequence data from two related populations. Thanks for listening. That's pretty much all I've got time for. If I've explained anything in too much or too little detail, feel free to ask any questions you might have at the Q&A, but I hope that I've at least given you some insight into the interaction of population biology, genetics, and conservation. So I guess that's it. Peace out.